All right. I am here in the stream. Hopefully we have some other people here as well. So today we're going to focus on talking about a little bit about the new processes, the new drop date, and chat about how to use the grade tool. And then we'll go through chapter 12, and I'll show you where to find the information about the test. Test is still under creation, but your book goes into a million times more detail than I want in some of these sections, okay? So don't stress out about all of the tiny pieces there. But I don't want you guys to worry about, do I have, do I remember whatever part of the RNA holoenzyme are? This is not biochemistry. The key here is that a big piece of what I'm trying to talk about is exactly how all the DNA manipulation in your cell leads to the DNA expression. Okay, so I do teach it a little bit different than the book, but I'm trying to explain it in a way that is understandable for you guys and not too heavy on the biochemistry. Again, we're mainly trying to understand how this relates to expression of genes. All right, I'll wait a few minutes until people get into here. There's a giant windstorm out my window, and everything's blowing around. My daughter's sandbox picked up and flew across the side. All the sand's gone now. Everything's fine, but somebody's trash can broke outside, and there's trash flying all over the neighborhood, so it's fun to watch outside, although I'm not very excited about cleaning it up. Hopefully everyone's safe. It was nice this morning. Hello, how's it going? It was beautiful until this windstorm came. You guys may be in a different area than me. I'm in Bowie. Bowie was beautiful in the morning. I dug up a garden bed. I put up some borders. I was like, maybe later today I will go out and plant some vegetables. Right now, if we go outside, everything is literally blowing around. The chairs on my patio are like across the yard. So it's very windy. It's beautiful to look at, but it's not really fun to play outside right now. At least my 3-year-old was happy, and then now she's sad that it's so windy. All right, so we'll get started. What I'll start with here is, this is new in course content, okay? I want to show you how to use this. I got as much grading as I could done last night, but I know that a few things were still due in terms of, yeah, super windy. So a few things are still due, but I'm going to explain kind of how to set this up. I want everyone to be able to do this on their own. Remember, you don't have to drop next week, even if you're failing. You have until the end of class. So this benefits you in 2 ways. Number one, you can, so if you're concerned about getting your money back and you are concerned that you really aren't making it, which a couple of people after the end of next week, I'll have an idea of where you are, and you might want to consider. A couple of people in this class may consider dropping. I'm not saying you have to, and maybe you're going to improve on the next test. But one of the keys here is that if you drop sooner, you get a larger refund. Now, if you're not concerned about that, you're concerned mainly about passing the class. I think that you can wait until you have more information, especially from the second test. And also, if you plan to retake this class next year, or take this class at a different institution, staying in the class as long as you can to get all the information so you can pass it better next time may be a good idea. But I'm going to show you what kind of some of the guidelines about where everyone is, okay? So this is what I sent to you. The key here is that there is 360 points left in class, okay? There is lecture exam 2, lecture final, topic quizzes, Those are the Blackboard quizzes we do. So there's 2 left of those. The journal. Class points is about 20 points left-ish. And then homework. There's 3 homeworks left. One of them's extra credit. So, I mean, I put it up there because you guys will probably still finish it. Case studies. There's 3 more of those left. Again, the one next week we're going to do in situ. I'm not going to share it beforehand, and you guys can do it after class, okay? So the final research project. So this is all the total points that remain. Okay. The key here, I have this little key up here. Fill out this field, don't touch this field, okay? Fill out the blue fields. So you put down that you expect to get 70% on lecture exam. I suggest you first start by filling out for all the lectures, for all the exams, the things that you've gotten on them before. So if you've gotten 100% on all the topic quizzes, you put a 1 there. You're good. But for the final and the lecture exam too, if you want to predict your grade, I suggest putting in what you got before and see if that will prevent you from getting the grade you want, okay? So I put kind of in the someone in the middle of the road in terms of points in the class, 240. So that's the current points. I don't think that any of you have exactly that number, but you're close. Several of you are close to that, okay? 240 if you got 70% on literally everything. The lecture exam, the final, the topic quiz, the journal, the class points, the homework, case studies, and the final research project. So this has a, don't disturb this because it has calculations in it, okay? Then you type in your current points of class. It's going to give you an expected score. Gives you a range now. You can see that C is 490 and above. So if someone's at 240 now and they stay at 70% for the rest of the class, they will get a C, just barely a C in class without much buffer. Potentially this person may want to consider improving in some other areas. Now, what if you're a bad test taker? Which I understand this is going to be your first online tests, like online long tests. So if you struggle with the test last time, or if you're struggling with some of the concepts here, maybe you're going to put a lower number here, okay? So let's say you get 60%. What this tells you, it doesn't tell you you will get a D. What it tells you is you have to improve the numbers elsewhere, okay? So let's say you're going to do all the topic quizzes. You've been getting 100% on them, so you put a 1. Let's say you're definitely doing all the class points. Let's say you haven't missed a homework yet. You get about maybe 90% on the homework, so put a 0.9 there. The case studies, you've been doing pretty well. On average, you get like a 90, so let's do 90%. And the final research project, maybe you're concerned about that, leave it at 70. And look, you still get a C, okay? So this is kind of what I want you guys to do, is think about where your strengths are and where you struggle, okay? So put a lower number over here if you struggle with some of the written parts. Put a lower number over here if you struggle with these, and then higher numbers wherever you've been succeeding. If you've been succeeding at the case studies, then give yourself as high of a score. Aim for the high scores in the case studies. You can figure out what is your worst case scenario. You know, I got a 70 on a case study once. What if I get 70s on all the case studies? I haven't been able to participate in all the class points. What if I get a 50% of the class points? And this is going to give you an idea of what you can fail on and what you can do instead. So if you have a higher number, if your current score in class is higher than that, then potentially this means that you're going to get a higher score overall. Now, I'm going to note that the case studies here, this 30 points, that doesn't include the one that was due yesterday. Again, if you turned in late, I didn't grade it yet. So if it's not graded, you can either assume what you're going to get. But the idea is that the class points don't reflect assigned but ungraded participation. If you did some of the participation parts, but I haven't graded it yet. I think I graded most of them, but then that would add to your score once I'm done grading it. Similarly, I didn't put in the assignment due on Wednesday, but that doesn't matter because it was due yesterday. So whatever you have now is your final score for that. And the case studies do not include case study 6 and 7 due this week. I graded most of the 6s. 7s, I think about half of you had it turned in, so I graded those. But if they're not graded yet, it's not going to be included in your score that you plug in here. So you may actually have about maybe 8 to 10 points more than that, okay? Again, play with that, figure out where your strengths are and where it's not. If you got a low score on the exams, start there. Put in your actual score that you got in the previous exams. Assume you're going to get in the future and where do you need to improve. And if you can't get to the grade point you want to get to, like you're 10 points below a B, 20 points below a B, right? Like you really want to get to a B, then you may have to think about how to improve your exam scores too. Does anyone have questions on how to use the grade tool? Wait for the stream to catch up. Okay. Fantastic. Again, please play with it. And if you are concerned about your grade when you plug things in, send me an email and I can send you a path to whatever grade you're interested in or tell you that path's probably not possible. I have told a couple people already, you're not going to make it to a B, but you're solidly in the C range and here's how you make it there. I've told a couple people, you're not going to make it to an A, but you're solidly in the B range and here's how you can keep it that way. Okay. Exam will open up on Wednesday and it will close on Saturday. This will give you time to, I know that being at home, guys have various obligations, so I'm giving you a Saturday as well to complete it. You don't have to complete it on Saturday. Feel free to finish it by Friday so you don't have to deal with it. But again, we don't have the drop date to stress out about, which I'm super happy about. Study guide is posted. Look in the study guide section. I definitely posted it last night. I posted the study guide. Okay, so look in your study guide. Yes, exactly. Use the blue to input what you think you're going to get. You can do worst case scenario. I'm really doing bad at this whole how do I build these problems about mapping. Say you're going to get 10 points lower than you got on the last exam. I'm not saying you will. I'm just indicating that maybe that's what you want to do, okay? And then you can figure out, well, can I survive that? Do I have enough buffer? And this will tell you that, okay? Again, if your case study 7 isn't graded yet, just put, 7 to 10 more points as long as you actually tried on it and turned it in last night. All right. Let's move forward. Again, everything is posted. I worked really hard last night to get everything posted. I'm running on fumes here. I have a 3-year-old I have to deal with at home. And I only have a nanny for a limited period of time. So I'm working my hardest, but sometimes you're going to see like a glut of things happen at like 10 p.m. Okay, all that to say. Let's go to PowerPoint. All right. This is too big. I need to make it smaller. Give me a second. Okay, here we go. All right. I think I have this set up properly, but double check. Eek. Nope, too big still. Gotta pull it up just a tiny bit so I can see it. Okay, sorry. I should have said that before. Okay. So, transcription, so we talked a little bit before. This is part 2. Again, part 1, we already talked about the elongation and other things. And we're focusing on RNA processing in this part. Oh, okay. If it's not there, my apologies. I swear I had posted it. I can post it right this second if you give me like 2 seconds. I swear I did though, guys. I was working so hard on it last night and then I must have forgotten to put it up. Okay, well, it's gonna be up now. Let's go to Blackboard. I'll put it up right now. I know, it's gonna be boring watching me work on a screen you can't see, but it'll be fine. All right. Content. Study guides. I swear I added it. Maybe I added it to the wrong one. I don't know. Okay, here you go. Study guide exam 2. Maybe I just didn't finish that because I definitely uploaded it. I don't know. It's here though. It's done. Let's go exams and study guide 2. Right. Okay, it's there now. I got it. Okay. All right, back to this. So we talked about elongation, and we talked about the, and then we started to talk about termination. We didn't finish it, okay? So let's talk about termination, okay? There was the row-independent termination, okay? So the row-independent termination is when there's these inverted repeats in the RNA transcript. Has these complementary sequences, okay? So you got TC, GGG, CGG, and then CC, GCC, CGA, okay? So you see those are opposites of each other. And so this is what, so it's going to come out with this. These two pieces make a hairpin, okay? And then there's this polyadenylation, poly-A tail, and it's going to make a poly-U there, and that breaks off, okay? Poly-U is weak. These are weak in it, so this long strain of weaker bonds leads it to fall off, and now you have that part, okay? So again, that's the row-independent termination. The row-dependent termination is a little bit different. It has to do with a small protein named rho, okay? So it binds to these C-rich regions, and RNA is threaded through this hexamer. It pulls rho towards the RNA polymerase that's building it. Once these touch, and so again, these are kind of these are programmed to touch it right when it's supposed to, like it's following it up, and it leads it to, again, these C-rich regions. Again, there's going to be a signal in there that tells it where to bond, and then it's going to follow it up and stop it at the right point. Okay. Yeah, I put it up. I thought I put it up last night. I think I just didn't press submit, but I definitely did it last night. Okay. It's organized a little bit differently, but we'll figure out where this, we'll look at the study guide after we finish this part. Okay. Rho and RNA polymerase together, that ends translation because it changes the shape of the RNA polymerase enzyme, and that breaks off, okay? All right, so let's just, from what you learned before, let's see if you guys remember. We have the promoter. Think about what describes the function of the promoter and what does not. What does the promoter do? That, and then what does it not do, really? Okay. Where is the promoter? What does it do? And what does it tell RNA polymerase? Okay, Fanya thinks it's a D. Everybody thinks it's D. Ebery and Afanya are just sitting next to each other. Okay, it is D because obviously the promoter is the start. Okay, the promoter is the beginning of the strand here. Okay, so it's a couple of, it's a little bit upstream. That's the promoter sequence. That's where it tells RNA polymerase to bind. It has that sigma that binds onto it, guiding the RNA polymerase to start. Okay. There you go. Afanya and Ebery, they're just sitting next to each other, like talking in a room. So it's because they're the only ones actually in the same room with each other. All right. So all the following statements about the process of transcription are correct, except there's a lot of reading here, huh? But let's remember about transcription and what the directionality and all that kind of stuff, okay? Yeah, take a minute to read. Any ideas? Okay, so Mavis says unsurely A. So the key is you think this is incorrect. Aha! There you go. It can't be B, says limit breaker Rana. Right. So we can eliminate some, right? Rana's going elimination here, okay? RNA molecule has a U. Therefore it's not E. We just talked about the promoter. This is correct. The RNA molecule has to be anti-parallel and complementary to the template DNA strand. Remember we talked about the template? Sorry, you're Rana. Limit breaker is... I've forgotten. I apologize. I can't see your faces. Okay, so limit breaker. Wait, who's that? I know everyone else's name. Kim, Angie. Right, IRS is the government. That's okay. It's not B. The RNA molecule is synthesized in 5 to 3. Same as the DNA molecule. Goes from the 5 towards the 3. That's the way it happens every time, okay? It's not D because the start site and direction of transcription are determined by a region off of the DNA of the template DNA strand. That's a promoter. On the bottom is where the RNA template, RNA polymerase will connect, okay? It's not D. Again, all the rest of them are correct. B is incorrect, okay? It isn't B because RNA is synthesized in the same direction as DNA, 5 to 3. You add on to the 3 end, that OH, free OH hydroxyl. All right. So remember what the sigma does, okay? Remember what sigma does. Who remembers what sigma does? It's that little protein comes along with RNA polymerase. What does it do? Only one strand that's transcribed, absolutely. It's always the template strand, and the promoter recognizes a sequence on the non-template strand, determining which one is the template strand. Is the opposite of where the promoter is. Okay, so there's a promoter segment that's recognized by, wait, what is it recognized by? So now we have, running into this question here, okay? The key, so I'll give you the answer here. The idea is that the sigma binds at the promoter, indicating then for RNA polymerase to start in the opposite strand, okay? Without that information, RNA polymerase would just start reading DNA wherever and not create a proper transcript. And that transcript would likely not end up being a proper, so this would be a specific sigma subunit bacterial RNA polymerase. All right. So we're going to be talking next about RNA processing. RNA processing is interesting, and I'm going to explain why. One of the key things I want you guys to get from RNA processing is exactly how DNA to RNA is not always, it's not a straight line. I think I put in to the new homework something about, okay, if the DNA is this, what is the mRNA, and then what is the protein? Now you can do that if there's no introns. So we're going to talk about exons and introns and the processing of the molecule to become actual mRNA that leaves the nucleus. All this happens in the nucleus. So pre-mRNA, after you cut out these introns, is further processed. We're going to talk about all its processing pieces, okay? All right. The key here is that mature mRNA, its transcript contains less nucleotides than the DNA template from which it was derived. Okay. So messenger RNA has 3 major regions. Okay, it has the 5' untranslated region. It has the protein coding segment. It has the 3' untranslated regions. Untranslated means it's not going to be part of the protein. Remember that we're gonna talk about this in a minute. Well, I'm not sure fully if we're going to make it there, but we'll talk for a while about this processing. But remember that when translation occurs, it always attaches to the first AUG. It reads it from 5 to 3. So it attaches to the first AUG, and it goes downstream from that, the ribosome. So there's a little bit ahead of it, that 5'. It starts on the 5 end and reads towards the 3 end. So you have a small segment, that's the 5' untranslated region, and then there's a translation stop, and then there's a segment at the end that's also untranslated, okay? So and actually, to make it, we have to put all these pieces in. So one of the key things here is the addition of this 5', what's called the methylguanine cap. So, and then also an addition of a poly A tail, okay? A is not going to build any proteins here, okay? Up to 250 can be added to the end of the mRNA. Remember the reason that we add things like, remember why we add things like telomeres on the end of our chromosomes. The poly A tail acts in a similar way, okay? So you have this methylguanine cap. It acts as literally a cap on a marker. It keeps it from degrading at the end that it would start to read from, and you have this untranslated region also to kind of buffer the coding sequence. And then the non-coding sequence at the end, that untranslated region, that's the poly A tail. All right. Again, first the cap is added. That cap can actively be added while it's still being built, while transcription is still occurring, okay? Again, it protects the transcript from being broken down. It's like a marker cap, or like if you're carrying a message in a tube, it's the cap on the tube, okay? It's just protecting it so that it makes it, and the end does not degrade because the 5' cap is pretty stable, okay? It also helps our ribosome attach the mRNA, and it knows where to attach because it detects that 5' cap. Next, the 3' end is cleaved. So you have this, in the pre-mRNA, you have this, what's called the consensus sequence. This is part of the untranslated region at the end. And A, okay? Then there's a couple of nucleotides here. Key here is that this is where it would stop, okay? And then you have this consensus sequence. And then the cleavage site is somewhere 11 to 30 nucleotides further. So you cut it off. And then, again, that U-rich sequence is due to the, remember when the row independent termination happens, it has that U-rich sequence at the end. So it cuts off the U's. Now it's shorter. And then it adds this poly A tail at the end. Okay, that is not part of the original sequence. That is, again, a protector. Kind of like a telomere. It's protecting it from degrading, preventing it from breaking down in its travel from the nucleus to the cytoplasm. So that helps stabilize the mRNA too, okay? So you have a buffered, important information on either side and protectors on both sides. Just different protectors because they have a different chemical makeup. All right, now the interesting part. RNA splicing of introns and exons, okay? So a lot of genes have introns in them. Kind of junk DNA is how sometimes it's viewed, but introns aren't junk. Think of them as these are different coding regions. It's one segment of the protein, then another segment of the protein, then another segment of the protein. Sort of, you break down building something into several steps. This is step 1, step 2, step 3 to make the protein. Okay? So the introns, they're intervening regions that are absent from mature mRNA. These do not change the amino acid code. But if you leave them in, they would change it, okay? RNA splicing overall removes these introns, these non-coded regions, and only leaves the exons, which actually produce the protein that the gene is trying to express. But again, these are more of this separates 1 from 2 and 2 from 3. Stick them all together and you have a full protein, but it's sort of a DNA organization way, okay? It gives the DNA some buffer, and it protects, think of these as also protectors of the important information. It allows for some, because mutations happen all the time, so if there's introns between your important areas, you can have mutations in there and not have any big issues. All right, so this is showing this is from your book, intron and exon sequences in various genes. So the numbers indicate the number of nucleotides present, okay? In mouse insulin, you've got this 2 segments and that are exons and then 1 small intron, okay? Rabbit beta globin has these 2 sections, and look at this chicken ovalbumin, okay? It has so many different pieces that are stuck together, and all of those add up into ovalbumin. Ovalbumin, that's the stuff, the protein that is inside of the egg. But again, there's more introns than there are exons here. So the question is, well, how does the cell even know which one to cut out? How do you think that works? Anyone have any idea how we actually tell what we're supposed to use and what we're not supposed to use? How do we tell there's only these chunks that goes into the chicken ovalbumin and not those chunks? Any ideas? See, Fanny's been listening to what a lot of these are, okay? There's promoter region, there's these end repetitive segments. Yes, there's markers that highlight where the introns are and where the introns start and where the exons begin. Well, okay. So interestingly, we have human G size, mRNA size, and number of introns, okay? Again, this is the gene size, but this is the mRNA, this red, it's 2 introns, okay? You see these big genes that are red. Look at this, dystrophin, 2,400, these are base sizes, okay? Base lengths. 2,400 bases long, only 17 are actually red. Has 79 introns. Again, one of the keys here is that introns are a way of, think of them as DNA protectors. If you've got enough space in between the important parts, then any errors are more likely to happen in the spaces. And then if you make sure you mark them, then they can be still functional. All right. So the gene concept here, we often think of If you read the DNA, you can immediately get the mRNA and you can immediately get the protein. So that's sort of a weird one here. You can't. And that's what part of what makes genetics so complex. You can't actually just look at the DNA to figure out what the gene is. Sometimes you have to look at the protein to figure it out, because we don't always know where the exons and introns are. We have some concept by watching it. We can do some interesting paying attention. But the DNA sequence, we have a lot of DNA in our bodies here, okay? So the gene includes the DNA sequence that codes for all exons, introns. Then there's also the beginning and the end of the RNA that's not translated. We have that promoter region. We have that terminator region. We've got those repetitive sequences and maybe make the RNA go into a loop and break off. All of those go into the gene itself. And we've got thousands of genes, okay? 20,000 gene sites in our DNA alone. That's a lot. So each of these has this pattern. The promoter is different all the time. Remember I told you the different promoters are in different RNA transcriptases. RNA polymerases, sorry. All right. So again, here you have the promoter and the coding sequence here. And then you have the additional 5 cap and the 3 poly tail. And you've got to add the cap, take out the introns, add the tail. You finally get mRNA and a polypeptide, okay? And they have some evidence, actually, that certain regions of DNA, only certain regions are included, okay? So, beta globin mRNA can be synthesized from mature mRNA transcripts of exons 1 and 2, okay? So basically, they make a beta globin mRNA, and in one of them, they put an intron in, okay? So if there were no intron, the DNA would hybridize with mRNA in a continuous strand. So we're hybridizing them, and we're seeing where it sticks. But if there's an intron there, the mRNA will stick on only the parts that it's transcribed. So they have this one where they take a one that they know doesn't have an intron, and then if they put the intron in, they know it has an intron. Then you see, oh look, that this part loops out, and you actually see that there's a part cut out. DNA is forced into a loop by the mRNA, which brings the 2 exons together. So you can see this loop here in the electron micrograph, okay? And so this shows you that the mRNA does have these uncoded regions, because the ones that they code for, they would create these loose loops that get cut off. Okay. So if they did that, then you can see how many introns there are in a single gene. Okay, so you have the 5N cap, and then there are these loops. So the loops represent the introns, okay? So those 3 loops represent the different introns that are not going to be read. Again, this is the RNA DNA hybrid, but they take the mature mRNA and they link it to a DNA that has introns and does not have introns. And you can see then from the mature mRNA where the introns are, because it will only stick and it's going to loop out those parts that are introns. So we can actually tell where the introns are using the mature mRNA to hybridize. We can also look at it for markers, but it's a little bit more complicated because we actually have to sequence it. We don't have to sequence it for that. Just cut out the piece that we know that the gene exists in, and see if you see any loops with the mature mRNA where the introns are already cut out. All right, so the splicing process I think is pretty interesting. It is a little bit technical. I'm not going to go through every single piece of it, but I want you to understand why RNA does what it does and how it does it, and then understanding some interesting pieces about alternative splicing. That gives you some weird expressivity issues here, okay? All right, so splicing has to be consistent and precise. Now, I mentioned alternative splicing already. I say always splicing in the same place for that gene product. Sometimes different gene products are produced, but for the specific gene product that they want to create, you have to always splice in the same place. So introns are marked by small areas of DNA. These snippets have actually already been transcribed. Remember that RNA polymerase II in eukaryotes recognizes different promoter regions. And these promoters are inside other genes. It recognizes a promoter sequence inside of another gene that is where the intron is. It makes these tiny little RNAs. They're called siRNAs. So those little siRNAs have already been transcribed from the specific genes here, okay? The siRNAs are complex with proteins to form small nuclear ribonucleoproteins. It's a mouthful. Geneticists are weird, and they call these SNRPs. Okay? Multiple SNRPs are formed into a spliceosome. Those SNRPs join, they link onto the different parts, and basically they make loops, similar to what we saw in the messenger RNA. They make loops and they chop the introns out based on where the different SNRPs are attached to the RNA. Okay? All right. So we have here exon 1 and an intron. The intron has a branch point, and then it has the end point, okay? So these little SNRPs come over and they stick on these little segments, okay? So the segment's slightly longer than this, but again, they're really small. And again, they are complexed with other SNRPs as well. And that forms this complex spliceosome that pulls it together and cuts it at the right spot, okay? Again, other siRNA combos are part of the complete spliceosome. And for this splicing, it cuts it at these points. But note that the same spliceosome could attach to two different sites and splice different sites. So that's an interesting piece about the spliceosome, is that there's a lot of SNRPs as part of it. And again, these SNRPs can cut out one intron, they can cut out multiple introns, they can cut out parts of exons, depending on how they attach to the RNA, the pre-RNA. All right, so this is where I talk about alternative RNA splicing, okay? So here you have the DNA in the RNA, okay? Exon 1, exon 2, exon 3, exon 4, exon 5. But sometimes different exons are left out of different proteins, okay? You have the mRNA for one protein. You got 1, 2, 3, 4, 5. In an alternative splice, exon 3 can be removed, creating a different protein with a different shape and a different function. A similar function. Note, often alternative RNA splicing creates similar proteins that do similar things, but not the exact same. Why would this be useful? In what case do you think the alternative RNA splicing would be useful for an organism? Why would you want to create 3 or 4 different proteins from a single strand of RNA, from a single stretch of DNA? Anybody? This is a tricky one. Why would we need to do this? What's the purpose here? What is the evolutionary purpose? What is the functional and the cell purpose? Everybody thinks genetic variation. That is one piece of it, is that having slightly genetically variant people in a group creates more genetic, just slight genetic variations that create multiplicity of organisms just from these tiny wobbles in the splicing of the RNA. What else? I'm going to give you an example. Let's consider something similar to a antibody. An antibody is a protein, correct? Until we encounter a specific flu or bacterial organism, we don't have the antibody. But then after we encounter it, we do. How? How do we make a protein that's not coded into our DNA? Ideas? How do we make a protein that's not coded into our DNA? So that's a good question. Actually, one of the ways that we can make various proteins, again, antibodies are a complex protein. Antibodies, what happens is that we have your body doesn't recognize, say, a virus when it comes in. After it comes in, our body attacks it and creates a bunch of different little proteins like this and tries them out on the virus. Basically, it says, here's a bunch of keys. Do any of these fit into the lock that would mark the, yes, our cells are marking the virus themselves, okay? So in terms of, say, the COVID, what they're targeting is they're trying to induce our body to, so we're targeting the spike protein. You've seen the little COVID pictures, this little ball spikes, okay? Targeting the spike protein, we're basically teaching our body to produce this protein that fits into the lock, marking it immediately when it comes into our blood as so we have an antibody to that. And that marks the virus and we kill it right away, okay? It's only when the virus is able to take hold that we're not able to fight it off. That's what happens. So if you have had any of the shots, basically your body produces a bunch of small proteins, finds one that sticks on the virus, and then uses that to mark all the viruses. But our body has to have the ability to learn. It has to be able to learn how to mark it. So being able to create a bunch of proteins that kind of are the same but slightly different shaped, very useful in terms of learning, changing the way that we express proteins based on their environment. And so adaptation to the acclimation to the environment via these alternative RNA splicing is one way that our bodies are prepped to survive and not get the same virus over and over and over again. So it's an interesting thing, okay? Again, the enzyme will cut a different splice signal sequences, leaving out 1 or 2 exons and creating a different protein, different shape in particular. Note that it never cuts any of the exons in half, okay? All the exons remain, but it leaves out one of the functional parts, changing the shape. Usually doesn't leave out like 3 or 4 of them, but it cuts out 1 exon here and 1 exon here. Different exons can be removed depending on the needs of the cell and using the same pattern as our body does to turn on and off genes, those silencer versus the promoter, the enhancer, okay? So again, sorry, this should say S in RNA. How does the cell know which to splice? SNRNAs that recognize a specific order of introns or exons to cut out are induced to transcribe. Basically, RNA polymerase II is told transcribe these marked parts. Don't transcribe these marked parts. And when there's more of those snRNAs, more of that type of spliceosome is going to be created, and more of the mRNA, the final mRNA, will be created. Now, it doesn't mean that you turn it off completely. Potentially, some of it will get spliced in one way and some will get spliced in another. But if we create the correct spliceosomes on a regular basis, you can create more of one type of the protein. So this gives us genetic flexibility and protein shape complexity. We can change the protein shapes little by little. Again, basically we're a locksmith. We're trying to find a key for the lock for various things we encounter. And once we find that key, we can produce a lot of that key. And that we need that protein shape complexity and flexibility to respond to the various stressors that our body's under. This helps individuals survive and thrive longer evolutionarily. So, again, if you have this this part here, okay, these transformers, again, these are other these are little things that tell it, okay, cut here or don't cut here, okay. These are other markers that come onto there. It creates a truncated protein versus a functional protein, and that can actually be different in between male and female. What it means is that the human genome can direct synthesis of many, many, many more proteins than would be expected due to its number of genes and length. So we aren't limited by the number of genes that we have. We can produce a lot of other things or be induced to produce them based on our environment. Up to 90% of all genes undergo alternative splicing. Conservatively, about 50%. But 90% of genes can undergo alternative splicing. Maybe your body doesn't, but it could. The result is complex polymorphs, where interacting genes get expressed differently in different people. Anyone remember where we last used the word polymorph in class? Where did we last use the word polymorph? Recognize that term? What did it describe? We were looking at an interesting polygene, something where we can't fully predict from parents what in newborns. What are we unable to predict? Just by looking at the parents, you can't tell what the newborns what will be. Anyone have an idea? Remember what polymorph meant? Sure, phenotype, that's more in the broad, sure, kind of, but specifically one trait that a lot of people want to know ahead of time, but they can't. Not like male or female, but like something particular. There you go. So Rana remembers eye color. We talked about polymorphs and eye color, okay? In that, sure, you can tell, is this person going to have blue eyes or brown eyes? But you can't tell what's the inner ring going to look like. How are the bands going to be structured? What shade will it be? And that's because of this alternative splicing creates these complex polymorphisms in our eyes. Depending on what set of spliceosomes are active in your eyes can actually change color over time as well. And that's because you can also react to your environment, and your eyes can get darker or lighter. And they won't change color completely, but they will be a little bit more green or a little bit more blue. Adding some of those little polymorphisms in with complexity of genetics. And so this is an interesting piece in how the evolution of eukaryotes has added these little wobbles. Talked about the wobble hypothesis already, that wobbling pairing ability to have that redundancy and be able to use multiple codes to create the same one. In chapter 13, we're also going to talk about the wobble. Okay. Again, complex polymorphs. Interacting genes can express differently in different people. Now, abnormal splicing in some genes has been linked to cancerous cells because you can splice out functional exons instead of just the exons that have to do with shape. So an interesting one, I'm going to be putting together a short thing. So this is from your book, actually. Let's see, where is it in your book? Let's go back here real quick. Okay. This is in your book. Let's see if I can scan this for those who don't have an access to this book right this moment. But the key here is that the gene muscular dystrophy is caused by an X-linked recessive disease. It's caused by errors in the dystrophin gene. I showed you the dystrophin gene earlier, remember? There's so many different exons and so many different introns as part of it. Errors in the dystrophin gene. Basically, it's built wrong. It's caused by this frame shift insertion error. It makes a stop codon. Remember that some of the codons code for stopping translation. It appears downstream of the error, and this means that you get a truncated, very short, and very not, it doesn't work very well, protein, okay? This happens in one of the exons, that frame shift error. Remember, though, that mRNA isn't read until it's full mRNA and not the pre-mRNA. Until introns and exons are cut out, this error doesn't do anything because the protein hasn't been made yet. So a new drug induces exon skipping and dystrophin transcription. So if the frame shift error occurs in one of the exons and shifts the reading frame, how would cutting it out fix it? How could skipping that exon help the protein? How could we fix some of the issues that are caused by Duchenne muscular dystrophy through skipping an exon? That's what I'm asking you right now. Any ideas? There you go. The stop codon doesn't appear. Now, it's not perfect. You can skip an exon that's broken. And the key here is that the exon is broken in a way so that it is a insertion error. So you have the exon slightly longer. If you skip it altogether, then you're still missing a couple of amino acids. But there is so many. And so what happens with this is that it goes amino acid, amino acid, amino acid. That error means that it goes amino acid, stop. You miss half the exons, and now you have a really short, really broken protein. Skip that one exon, and now you don't have the frame shift error, and the rest of the amino acids will work. You have a longer, albeit slightly differently shaped protein. The error-ridden segment is spliced out. It results in a more complete protein that can be produced by your body. And this is a form of inducing alternative splicing of a protein, which is pretty cool, but weird, and maybe in some ways a little bit concerning in ethics. What if this doesn't work the same way in many people? What if we induce it to skip the wrong exon and then they get worse? There's obviously some stresses here, but the idea is that Duchenne muscular dystrophy is a deadly disease. Men who have it, women can't have it. The double recessive actually dies in utero. But men who have it, they don't live past the age of 18. So even having some functionality return is a better alternative than slowly wasting away. All right, so what I'll do is I'm going to scan that and upload it, and you'll answer the questions that are part of it, okay? So this is a participation activity. I remind everyone for the participation activities to make sure that you are using your own words to describe what you're learning, and it's going to have you look up some stuff outside of the activity. Again, make sure you're not copy-pasting from those and just summarizing what you learned. You don't have to create a super long thing, just answer some of the questions, talk about the ethicality, the they did an accelerated approval because people were suffering so badly from this. And then, so then you talk about, can some of these drugs be good or bad, or how does this work, okay? But it's an interesting one, right? All right, here's a summary overall of the RNA processing that we've gone through. The promoter region, the reading part, the cutting out of so first we do the cap and the poly A, then splicing through the spliceosome, and then you have the mature mRNA that's ready to leave and go to be translated. Yes, it's going to include all the information needed, okay? I know that some people don't have access, some people do have access, so I'm going to scan that one page and then also include the, I think there's 2 links that you have to investigate as part of it, okay? All right, so alternative 3' cleavage sites. Remember where they break? What does that result in? Remember what a gene is to help you answer this question. Remember that this has to do with the mature mRNA, and those different mRNAs, that's the alternate cleavage sites, okay? Cutting out the introns or exons in various parts. Multiple mRNAs are different lengths. So all the following are part of mature mRNA in eukaryotic cells, except what part is not part of the mature mRNA? The one that leaves the, there, okay. You guys are more sure about this one. We just talked about it, right? Okay, introns, yes, introns. All the following are part of mature mRNA except introns. Okay. So here's a concept here, okay? We have 4 possible RNA products of this gene. So we're going to figure out what may have caused each mRNA. Again, these various mutations happen in various people, and that changes the way that the exons and introns are induced and such, okay? So let's consider, again, this would be something like you would do in your homework. I looked for a question like this. I don't think it shows up in your homework, but you may need to answer something similar to this in the exam, okay? So this one, you have exon 1, exon 2, exon 3, okay? Number 4 has exon 1, exon 2, and exon 3. So let's start here. That has to be the no mutation in any splicing signal, okay? You see that all the exons are there. So now you don't have any different splicing possibilities for that one. This one has intron 2. So what would that mean there? It has intron 2, but it doesn't have intron 1. So Afania says E. So and that would be true. So the idea is that if the splicing signal sequence is wrong, you don't have the proper signaling protein, that SNRP sitting on there, then it's not going to cleave there. And so you leave intron 2 in, which again will create a different one. Generally, in terms of the ones that alternatively splice, this is talking about ones that are leaving introns in, okay? So alternatively splice usually take out different exons, but this one is leaving in an intron, which generally is not translated and would produce a not proper protein. All right, what about when there's no mRNA produced? What could have caused that? Which one of these would cause no mRNA at all? You just don't have it. It's not even transcribed. Okay, C, right? If the gene's promoter sequence, remember that's part of the gene. If the gene's promoter sequence is broken and the promoter, you don't have sigma attached to that specific promoter sequence to create the mRNA, then it just doesn't create it. So therefore, that part would be a absence of a specific thing. This doesn't have to be bad or like deadly or whatever. Recognize that people who are lacking, say, maybe they have a promoter problem for something that creates pigment in hair, the depositor protein for pigment in hair. Well, we just have blonde hair then. So again, this isn't always deadly. The idea is that this can be a mutation that can pop up in various populations. So we talked about that blonde hair, okay? All right, number 3, both the introns are in it. Create a really long protein that's not going to make the same amount of sense. Okay, so that has to be obviously the last one here. Signal sequences both intron 1 and intron 2. Okay, so you don't have any of those SNRPs attached, so it doesn't know to cut out intron 1 and intron 2. Okay. All right, so that's all I have for that part. The last piece left, I would like to get that in by, so we don't have a ton more to cover. I'll cover things I'll just introduce the concept of translation. You guys know what translation is, right? You've gone through some of this. I'm going to go through really short. I probably won't finish it today. I just want to introduce it. And then we'll finish it up on Tuesday. But translation will be in there. Note that I don't need you to remember all the translation pieces. I think that your chapter 13 is like, here's this one gene, one enzyme. Here's all the processes for bringing through the translation, all the different segments, and show these. I don't think that's necessary. Understanding one gene, one enzyme is good. And understanding that, one gene, multiple enzymes based on this alternative splicing, sure. But the idea mostly is that one enzyme comes from one gene. Okay? But don't stress out about all of these various, here's all the elongation, and here's all the variation, protein structure, and biological diversity. Remember all the functional groups? That's biochemistry. I just want you to understand the basics of how a mRNA is read by the RNA. Okay, so let's go to number 13. Open. Okay, you're here. There we go. Translation. Okay. Again, it's short, and we're not going through protein structure and the variations in protein structure and how that influences genetic variation. We've talked about that a little bit with transcription, and that's enough. I think we're good. Okay. Oops. Goodbye. I gotta switch out. This is doing a slideshow, otherwise it's gonna take up my entire screen. I don't want that. Okay, all right. I'm going through 13.1 through 13.4, just basic genetic code and translation. Okay, we are not going through protein structure. That's too much. We're not biochemists. If you're taking biochemistry right now, that's fine, but I get exhausted by this. So anyways, we're not going to go through a lot right now. But The basics of translation is the last part here, okay? You have the mature mRNA. Whatever mRNA is created, okay, that mRNA is going to turn into a specific protein. So converting the language of nucleic acids into the language of proteins. So there's obviously this triplet code, okay? I'm going to revisit that because we visited it before. Then I'll talk about the actual process of translation on next Tuesday, okay? But the triplet code, important to know that each of these codons in the mRNA is read as a word, okay? We already talked a little bit about those frame shift mutations, the frame shift mutations indicating that these different codons are going to, if you put in one, then it's going to shift everything over, okay? But triplets in RNA are called codons, and there's 64 possible codons, and 3 of those are stop, which do not code for anything, and then 61 are sense, including 1 start codon. That start codon does code for an amino acid. It always codes to the same amino acids. Okay, so here's again, we went through this before, but here's the stop codons, okay? Translation stops when these codons are reached. We talked about that as part of the issue in the gene muscular dystrophy is that a stop codon was reached too soon and the entire protein is not translated. Also, it's translated wrong because it's changing the frame shift, so it's putting in the wrong amino acids. Sense codes encode amino acids. AUG is always the start codon, methionine, and the termination codons are these 3. Okay. Okay, 64 possible codons. Again, you've gone through a lot of those. The genetic code is degenerate. Means more than, each amino acid may be specified by more than 1 codon. This is the wobble hypothesis. The first 2 are the most important. The last one is not as important. And this helps a little bit with how tRNA and mRNA interact, okay? So tRNA brings the correct amino acid to the ribosome. Okay, once mRNA is in the ribosome and the ribosome clamps on, then you have this anticodon that's complementary with the base pair. Okay, so this is the codon. The codon is always used as the correct transcript, right? The anticodon is just indicating that this serine goes with UCC. Okay. Each type of tRNA attaches to only a single type of amino acid, and most cells have 30 to 50 different tRNAs. There's a lot of amino acids. Some amino acids are carried, there's 20 amino acids, so some amino acids are carried by more than 1 tRNA, and that's because this is the redundancy of the degeneracy of the genetic code, okay? So, there's these things called iso-accepting tRNAs. Different tRNAs accept the same amino acid but have different anticodons, and that's because anticodons can match to the same one. I wanted to go to the wobble one here, okay? So here we have this anticodon. The pairing at the third codon position is relaxed. The G in the anticodon can pair with C or with U. And that's normally not how they bond. I wanted to show this to you because this helps a little bit, because remember that the first 2 are generally the most important. And so this allows for 1 tRNA to bond to either one of these positions, but the serine still gets there, and the serine is still coded for by UCC or UCU, okay? That's why you have the wobble part. Okay, so here's the anticodons for the various things, and then there's no anticodon for the stop codons. Instead, this is the detached signal. Instead, it attaches this sort of like a water molecule, which breaks it off. Okay. You should learn most of this in 1140. I don't want to overwhelm you with like old information, but this is the main thing I wanted to get. This wobble hypothesis, understanding that the degeneracy of the code allows for some of these to match. Even though the codon's not quite, it can match at G or U, because again, the first 2 are the real code, and the third one, well, this TNR can bond there, this tRNA bonds there, but it's bringing the serine either time, and UCC and UCU bonds either. Okay. So again, these are identical tRNAs, slightly different codons. Same concept here, okay? G and C can normally pair, and it's always GAG here, but it can also bond with U, and that's the wobble pairing, and that's because it's not a true pairing. This is working with the degeneracy part, okay? All right. So I'll leave it at that. Let's take until 3:30, as I mentioned. I don't anticipate it's going to take too long to go through this. Mostly your homework this week was on replication and understanding the different types of replication, and making sure that you understood the various parts that went into that. I posted a couple of extra videos to go through. I can post a video for translation too. It's not going to be too complex. Okay. Does anyone have questions before? I posted up that permanent link to our recitation sections. You can go back to it. I'm going to see if I can post it up. Let me see if I can make sure you can scroll back to it, but I'll make sure that it's available outside of the announcements as well. I'll put it into the recitation area. Okay, so into the case studies, and you can click on it through there. Okay. So, questions about this segment? Again, don't stress out about all the complex biochemistry that your book goes through. This is for various types of genetics. We are not doing molecular genetics. We are doing genetics. So this is Introduction to Genetics. This can be a molecular genetics book too. But the key here is that we're not looking too heavily into the biochemistry piece. Understanding, though, how translation and expressivity interact. And kind of how the proteins influence it. Okay. Any questions? Well, we'll finish up translation. It's not that big. It's not that long. And then we'll go through the study guide. Let me pull up the study guide real quick in case you wanted to see it. My most recent thing I worked on yesterday. I worked on a bunch of stuff. See, there it is. Last night, like 9:30, finished this up. Okay, so this is, I organized it differently this time. So this is based on someone else's, but I made sure that I was only including the parts that we actually are going to be tested on, okay? But these are the chapter 7, probably the toughest part. It's going to be part of this. Obviously, it will be a big part of this exam. It's going to be a smaller part of the final because that's not all the final's going to be on, okay? But just make sure you understand some of these pieces, okay? And then there's the bacterial recombination. Very short. I truncated it because I'm going to revisit bacteria when we talk about genetic recombination and DNA tech, okay? RNA and DNA structure, DNA replication, chromosomal structure, transcription, RNA processing are all the things we've gone through in the last few weeks. Okay. And then the translation and protein building, we've gone through most of this already, so it's not going to be too much. Okay. And then we'll apply the wobble rules as well. Okay, great. I will see you guys shortly. Again, remember I put up that permanent link that's going to still work, still up on my Zoom. I'm going to press it, and I'll be back in 15 minutes, okay? Again, it shouldn't take too long to go through. There wasn't any calculation ones on there. I think there was one or two longer ones where you had to like organize it, but shouldn't be too difficult. So maybe we can have a slightly shorter day today. Like we can have a short. I could try to upload everything before I go to sleep tonight, so we'll work on that. All right, I'll see you guys in a few minutes. Hopefully. Again, if you're having trouble with Zoom, I will record it this time. I forgot I'm going to press the automatically record for this session, so I apologize for not recording it on Tuesday. All right. Talk to you guys soon.